sini, kita dapat melakukan pembuatan tablet yang dimulai dari proses pembuatan granul hingga pembuatan tablet, serta evaluasi dari granul dan evaluasi dari tablet. Yuk, cek laboratorium selanjutnya. Di laboratorium ini, kita bisa mempelajari berbagai macam bagian tanaman obat loh. Dan di sini, kita juga bisa melihat gambaran struktur dari tanaman obat-obat tersebut. Lanjut yuk ke laboratorium selanjutnya, yaitu laboratorium steril. Nah, laboratorium steril terdiri dari 3 area, yaitu black area, gray area, and white area. Ini adalah black area, tempat sterilisasi alat dan bahan. Yang ini adalah gray area, tempat sterilisasi diri. Yang ini adalah white area, tempat kita melakukan pencampuran bahan dengan menggunakan alat laminar airflow. Selamat datang di Apotek Pendidikan. Di sini kita dapat melakukan simulasi konseling mengenai informasi pengobatan dan penggunaan obat-obatan.
Mohon ditunggu, saya siapkan dulu persis. Mungkin itu aja. Terima kasih Bapak, semoga lekas sembuh.
Beberapa fasilitas yang ada di program studi Farmasi UTAM 45 Jakarta. Untuk info lebih lanjutnya, yuk kita dengerin penjelasan dari Prodi Farmasi UTAM 45 Jakarta.
Assalamualaikum, kenalkan, saya Apoteker Rabima Master Farmasi sebagai Ketua Program Studi Sarjana Farmasi UTAM 45 Jakarta. Seperti kita ketahui, program studi Sarjana Farmasi merupakan program studi yang mempelajari tentang obat-obatan. Di sini kita akan belajar mengenal senyawa obat dan sintesis. Belajar meracik, memformulasi, dan membuat obat yang sesuai dengan standar pemerintah. Tidak hanya meracik dan memformulasi, di Prodi Farmasi kita juga harus mengetahui bagaimana penggunaan obat-obatan tersebut secara aman dan legal. Mahasiswa kami diajar oleh orang-orang yang profesional di bidangnya. Pembelajaran di Prodi Sarjana Farmasi UTAM 45 Jakarta dilaksanakan secara daring, online, dan tatap muka atau offline. UTAM 45 Jakarta mempunyai program beasiswa untuk mahasiswa kami yang berprestasi. Untuk wadah kreativitas mahasiswa, kita mempunyai HMPS dan program UKM. Fakultas Farmasi UTAM 45 Jakarta telah bekerja sama dengan universitas-universitas ternama baik di dalam maupun di luar negeri. Secara rutin, program studi sarjana farmasi mengadakan event-event tingkat nasional dan internasional seperti lomba pasien konseling event atau PCA dan seminar-seminar farmasi internasional.
Jadi, tunggu apa lagi? Yuk, daftarkan diri kalian di UTAM 45 Jakarta.
Getting tomorrow, future today.
Oke, selamat siang.
Siang. Siang, Ibu.
Siang, Ibu.
Siang, Ibu.
Why? What happened with my screen? Hai. Oke. Dr. Mansour, how are you? Jam berapa kita mulai, Lulu?
Jam 1,
Semuanya
Bu.
sudah ya?
Jam 1, Bu.
Betul ya. Berapa menit lagi? 2 menit lagi ya?
Iya, Bu. 1 menit lagi, Bu.
Oke. Sudah bisa kita mulai?
Baik, Bu.
Oke, silakan.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I am Feni Angganastiti as MC at this event, the guest lecture today. Guest lecture today is about cancer. The Honorable Dr. Mansoureh Nazari as speaker. The Honorable Dr. Apothekar Diana Laila Ramatila M.Farm as the Vice Chancellor. The Honorable Apothekar Rabima SSE M.Farm as Dekan of Pharmacy. The Honorable Lecturers of UTE 45 Jakarta. The participants of the lectures, all participants. On this nice occasion, let me deliver the structure of the event today as follows. First, opening. To the speech from Dr. Apothekar Diana Laila Ramatila M.Farm as the Vice Chancellor. 3. Reading of the speaker curriculum vitae. 4. The guest lecture which will be delivered by Dr. Mansoureh Nazari from Iran. 5. The question and answer session. 6. Door prize and closing. Before we start, let's open this event by saying Basmalah. Bismillahirrahmanirrahim. Next, we invite Dr. Apothekar Diana Laila Ramatila M.Farm to give a speech.
Thank you, dear. Assalamualaikum warahmatullahi wabarakatuh. Everybody, I hope everybody is well, fine also. I think I have to say hello from Iran because of I saw some of Dr. Mansour friends from Iran. Hi, how are you? Today is the first time for us to have Dr. Mansour as a speaker to give a guest lecture in our university for our students. We are very happy and we are very excited, of course, to hear the sharing knowledge that Dr. Mansour will give to us. First of all, definitely I have to say thanks to Dr. Mansour for the time, and I also want to say thanks for all the participants who also joined in this Zoom webinar meeting in this afternoon. Because of the COVID-19. So now everywhere in the global, the method of sharing knowledge is different, not only offline by coming to see the seminar directly, but now we can see each other from our place, like you from Iran and us from Jakarta. And I'm very happy also because of the topic is about cancer, one of our subject in pharmacotherapy. And I told to my students also, it's very beneficial for you to hear and to ask the questions related to this topic to Dr. Mansoureh. And hopefully this sharing knowledge can give beneficial and the students before maybe they don't know much about cancer, then after they hear about this guest lecture, they will know more about cancer. And that's from me. And me as a representative of pharmacy faculty, maybe the dean of pharmacies cannot be here due to some reason. And I'm the one who will open the guest lecture, this seminar today. And I want all of the students will be active. If you don't know about the things about cancer related to cancer, you have to ask the question because she's very expert on this. She has she has a patent, and she also was a professional in the industrial. So she's the right person for you to ask about this thing. So I think this is a short speech from me. Thank you very much for your time. And hope this seminar will going well. And thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thanks to Dr. Apotheker Diana Laila Ramatila M.Farm for her speech. Next, we'll be greeted by a moderator to Gena Nafta Araminda. Time and place are yours. Gena.
Okay, thank you. Good afternoon, ladies and gentlemen. My name is Gena, and I'm a pharmacy student of UTF 45 Jakarta. And I'm your moderator for today's guest lecture. First, I would like to introduce our great speaker for today, Dr. Mansoureh Nazari from Iran. Okay, first I would like to read her CV. Her expert area and interest are pharmacology, biochemistry, biotechnology, cancer research, drug delivery, drug development, food industry, and chemistry. Her academic and professional qualification: in 2009, Bachelor of Science in Biology at University of Guilan of Iran. And 2016, Master degree in Pharmaceutical Sciences at University Saint Malaysia of Malaysia. In 2019, PhD in Pharmaceutical Sciences at University Saint Malaysia of Malaysia. Her industrial experiences: first, research officer in scaling up of developed product in nutraceuticals as the MBHD Kedah, Malaysia, 2013 until 2019. Second, research officer in herbal formulation lab in Eman Biodiscoveries as the MBHD Pulau Penang, Malaysia, 2016 until 2019. Third, R&D manager in Dextrum Industries as the MBHD Bukit Mertajam, Malaysia, 2019. Her other experiences: first, lecturer English language in 2009 until 2011. Second, manager and founder of Computer and Accounting Institute, Madaras, Guilan, Iran, 2009 till 2013. And third, Deputy CEO of Handwave Carpet of Sosanechel Cherak Company, Ras, Guilan, Iran, 2008 until 2013. Fourth, founder of Training Program of Managers for Private Institutes, and 5th R&D Manager of Textchem SDN PhD. Before we move on to the next session, I would like to remind you our rules. Please listen carefully. First, all participants, please to open the camera. And second, as long as the speaker delivers the lecture, participants are not allowed to interrupt. And third, the question and answer session will be open after the lecture. Okay, we are moving right along to the core of this event. Please welcome Dr. Mansoureh Nazari. To Dr. Mansoureh, please.
Yeah. Can you hear my voice now?
Yes, now we can hear you.
Okay. Yeah. Yeah, almost a few minutes I talk to myself. Okay. Okay, good morning everyone from Iran. Although I know that it is noon over there. I'm Mansoureh. I hope to have a good experience and sharing some information with each other. So hope to be useful for everyone. So I cannot see my slides. I don't know, should I share myself or it will be shared from that side?
Nadia. Okay.
Yeah. Yeah. Okay, thank you. So I think should be, yeah. So it means that I cannot control the slides, right? If it would be like I could control it myself, would be easier, you know? Is it possible?
Yes, doctor.
Is it possible that I control the mouse and then because I need to point out some parts, it will be easier for me to control the mouse by myself. Is it possible?
No, I think it's not possible unless if you
It
are
is
the
not.
yes who share this slide.
Uh,
Not if you want to control, maybe.
oh, okay. So it's not.
Yes, not.
Yeah, is it possible I share myself?
Yes,
It
it's
will be
possible.
easier
It's
because.
possible. Kasih Dokter
Okay.
jadi hostnya ya. Mungkin ini
Yeah.
share host and give the host to Dr. Man.
Yeah. Yeah. Thank you so much. Oh, I want to use this time to thank all of you, especially Dr. Diana for introduction and for nice hospitality. It was really nice to me in the early morning by Saturday. I like that. Thank you. Still disabled. 밑에 비용 매설해야 돼요. 네. Hồi Nazari 이제 보러 온 말. 세단에요. Okay. Hello.
Hello.
Oh yeah. Yep.
Nadia, maybe stop screen first. Yes. Yes.
Yeah. Okay. Now is easier. Okay. Now it's easier. Okay. Is it clear for everyone? Everyone can hear my voice and see my slides, right?
Yes.
Yes, Miss.
Okay. Okay. In the name of God, I'm a kind of PhD graduated pharmacologist. I did my master and PhD doing research in this field. So that's why, for this lecture, I decided to focus on some cancer research strategies, especially useful for young scientists. And then at the end, about therapeutic strategies that has been employed until today about this disease. So we move forward. The contents that I planned for today's lecture is first of all a briefly introduction about cancer. After that, about angiogenesis, and I will say step by step what is the relationship between them. And then after that, about some analysis in vitro cancer studies. Furthermore, about in vivo cancer studies, which is a kind of preclinical studies. And then after that, briefly about different methodologies in cancer therapy. So first of all, what is cancer? As most of you know, cancer is just abnormal cell division, which will be out of control and can be mediated by a cascade of factors
Kalau
that causes
Universitas
death.
17 Agustus
It
tuh ada
sometimes can
berita ya.
be
17 Agustus Jakarta. Universitas.
okay.
Please mute.
Sorry. Okay. Can I continue? Yeah. Okay. So I just was telling them what is cancer. Cancer is abnormal cell division mediated by few factors, numerous type of factors, which can be due to some genetic modification, which is called as mutation, gene mutation, or sometimes which modifies the expression of proteins, and so many events that can happen, and finally we will face the disease, which is almost the reason of 50% of death in the world, that is cancer. Okay, but even though some modification in morphology of cells in the tissues can be the reason of cancer, but not any modification in morphology can be cancer. I mean, it won't be identified as cancer. As you can see here, normal cells are arranged in the tissue. In morphology will be something like the photo on the left side. But after, is it possible to mute other
All the
microphones once
participants, please
I'm
mute.
giving?
Okay. Sorry, host, please mute all. Host, please mute all. Host harus ya harus selalu cekatan. Penny, mute all. Jadi setiap yang baru masuk.
Sorry, doctor. You are muted.
Host unmute yang Dr. Mansur ya, I think. Dr. Mansur termute. Di unmute Dr. Mansurnya. Dr. Mansur, sorry, your voice is mute.
Coba tuh dibayangkan ya.
Okay, do you hear me now?
Yes, now here.
Okay, wait. Let me go ahead. So everything is fine now. Everyone can hear my voice clearly.
Yes, yes.
Okay. So I will continue from the third slide. Okay. So as I mentioned here, so normal condition, the cells of the arrangement of the cells in normal condition will be something like, as you can see here in this slide, the left side. So in hyperplasia is a kind of condition which the number of the cells will increase, but it doesn't cause any modification in the morphology of the cell. So it won't be recognized as cancer. So so it's not like that if you see any additional number of the cells, so definitely it won't be the only reason for calling as cancer. But sometimes, as you can see in dysplasia condition, even though the number of the cells is increased at the third one, even though the number of the cells increase, but the morphology of the cells also has been changed. In that condition, it is more risky to be converted to cancer. So sometimes it won't be treated, but sometimes it needs to be treated. So dysplasia is a kind of severe condition compared with others. If I want to make an example here, is something like mole, that it has the potential to convert to skin cancer like melanoma, but it is not 100% that we convert to melanoma. It's something like that. We call it dysplasia. And the worst condition among all is cancer. In cancer, even though the number of the cells will increase abnormally, and also the morphology of the cells too will modify. So, and then the cells gradually, by being progress in the cascade of cancer progression, will move towards other tissue, which we call as metastasis, and then that's actually the last stage of the progression in cancer. It needs to be considered for serious treatment. Okay. Okay, different types of cancer. How it will be called depends on the source, I mean the origin of the cancer that's going to happen. For example, in sometimes it will happen in soft tissues or bones. So it will be called as sarcoma. As you can see, it may happen in lymph vessels. It can happen in blood vessels, fat, nerves, and others. Sometimes it will be called, you will hear like, okay, for example, this condition is just carcinoma in situ. It is not a last stage of the cancer because it's in situ, because it won't invade out of the boundaries that from the original cancer formation. So since it doesn't move it won't move from the original side to other side, it will be called a carcinoma in situ, and it's easier to be treated rather than some cancer, which you can see here, is too big, and then it will invade towards other tissues. So the treatment here will be quite serious, and it won't be easy for 100% survival. So, okay. here in this slide, I want to show you in a schematic view that how the drug can affect to convert the tumor to normal conditions. That's a kind of, different stages the drug started to produce. Here, for example, we start from this position at the first level. Some DNA mutations can change normal DNA to modified DNA, which is the kind of cancerous cause DNA. So if we be able to repair those DNA mutations, we will be able to convert again mutated DNA to normal DNA, and then ultimately can cause normal tissue, normal cells, and the patient will survive. So if this is step we couldn't do any treatment, we will go ahead on the next stage, which is deregulated signal transduction pathways. So if we have a designed kind of molecules as a treatment to inhibit signalase molecule, in that case, deregulated signal transduction pathway will convert to regulated signal transduction pathway. And if even we couldn't design such treatment, if we couldn't achieve such achievement, actually, we will go ahead to next step. Next step is, for example, the phenomenon of deregulated signal transduction pathway will be overactive or permanently active process. It is some expression of the proteins or blah blah will be overactive, overexpressed, and that's the thing that will be blocked by a specific medicine, and then ultimately can cause that overactive or modified process to temporary active process, and will ultimately reach to the normal condition. Okay, we go to the last stage. We couldn't inhibit the abnormal procedure of cancer procedure until here, so we reach to increase proliferation rate and decrease apoptosis rate, which ultimately can cause high cell number, high modified or abnormal cell number. So in that case, we don't have any other choice. We can only think about the drug that can kill the cells. That's how that our conventional what? That's how that chemotherapy drugs affected and cause kill the cells and try to convert the patient from abnormal condition to physiological cell number and normal condition. Okay, just in case everyone can hear my voice and see properly the slides.
Yes, I can see the slide.
Okay. Okay, we continue then. Okay, I want to talk more and take you deeper in the process of cancer progress, formation, and so on. As I said in earlier slides, I told that how we call a new formation of cells a kind of cancer. When the number of abnormal cells increase abnormally out of control, we call it cancer. Okay, but okay, what is wrong with increasing the number of the cells? Those cells that are increased in number, they need nutrients, they need oxygen, but due to normal condition of blood vessels, they cannot survive. So they need more blood vessels in order to get nutrient and oxygen. So what they do, they will try to excrete some signals from themselves. Actually, as you can see here, for example, some tumors are here. They cannot reach and get enough supply because some of them, they will finish the nutrient, and the rest of new formed cells, they don't have anything to survive. So they try the best for their own survival. What they do, they induce some signals that, for example, we don't have enough oxygen. Like hypoxia factors is actually due to this. They don't have enough oxygen, they don't have enough nutrient. They induce hypoxia factors. There are some molecular factors that they introduce it to surrounding blood vessels, and they inform that we need blood vessels, we need oxygen. In that case, When this happens, this new blood vessel will start secreting, after receiving those signals, will start secreting some factors which called as growth factors. Okay, those growth factors will start to permeate from the blood vessels to outside. In that case, they try to invade out and then degrade actually the mouse doesn't work sometimes. Degrade the basement here using some metalloproteinase. These proteinase will actually digest those basement membrane and let the epithelial cells here to grow and then migrate from here to outside. This calls as invasion because it's trying to invade out of the available blood vessels. And then at this step, after invasion, migration will happen. It moved out, and then it will be differentiated as a kind of, which is happening here, and a start. You see, that's the start of primary new blood vessels. So imagine that such origin of new blood vessels, each one will be bigger and bigger and will move towards those new cells that need nutrients. That's how those new cells will be surviving here and use their as you know, they will make their own blood vessels to be survived and go out of hypoxia condition. So as I just briefly told you here, you can see the angiogenesis as a kind of making new blood vessels from available blood vessels is a kind of reasonable reason for to continue being survived and continue and progress the cancer. Okay. What is angiogenesis actually in the body? It is something like, as you can see here, I hope that everyone can see this part of my slide. Angiogenic is a kind of switch, you know, between activators and inhibitors. Because our body automatically, it has both sides molecules. It has activators and at the same time it has inhibitors. Inhibitors is something that will stop something like cancer to happen. But activators is something that it will precede the phenomena of angiogenesis. It is really actually reasonable. You know why? For example, if I face some wound, if I make some wound in my hand or any tissue of my part, my body, so what will happen? Switching the angiogenic towards activators will help my body to start proliferation and repairing my body, those parts that has been injured. So for wound healing, angiogenesis is something good for my body. But at the same time, when it is going to be killing me, like phenomenon of cancer, it should be stopped. Otherwise, it will neutralize the cancer cells and will progress in the process of cancer. So it should be stopped. So normal physiological condition, it depends on how the balance between angiogenic activators and inhibitors will be stable. So the stabilizing condition between these two will produce actually the safety for human being. I hope you enjoy this part because it's my lovely topic, actually, angiogenesis. So, okay, angiogenesis is not just one phenomena, you know, it is not once at a time. It is a cascade of events that continuously happens. So, for example, if first step doesn't happen, the second step won't happen. If doesn't reach to the last stage also, it won't finalize the producing the new blood vessels, so nothing will happen. So it is a kind of cascade of events that continuously happen in the body. Depends on whether disease condition or normal healthy body condition. So I'm focusing here on cancer, how angiogenesis can affect cancer progress. Okay, so Here, as you can see, for example, imagine some new formed cells after division, cell divisions, which we expected to be cancerous, and they don't have enough nutrient and oxygen. What will happen? Hypoxia will happen. They don't have oxygen. They say, oh, we need oxygen, we need nutrient. In that condition, then HIF factors or hypoxia inflammation factors, those factors will be induced. So those factors will affect the proliferation, will give order to the proliferation step of epithelial cells. They will say, okay, epithelial cells start proliferation, and they will divide in the valves of the blood vessels, available blood vessels. After that, what will happen actually? Using some metalloproteinase, the walls of those epithelial cells, the walls will start to be degraded, and those new formed cells will move forward, will invade outside at the step called invasion. Okay. After that, moving a step of those new blood vessels towards finding a new place for in order to form will be called as migration. So migration will happen. After that, this, as you can see here, these cells are quite primary form of the cells that just developed, but it hasn't been differentiation. The differentiation will happen at this stage. As you can see, the blood vessels formed here at the stage of tube formation. So different primary stages of angiogenesis. First of all, proliferation, then invasion, migration, and tube formation. That's how new blood vessels will form here. Okay, one interesting thing here I want to tell you is this, that these new blood vessels that form here, actually, they will move, right? They will move, but towards which part? They won't move towards, for example, the normal cell line here or probably here. They will move forward towards the cell lines that induces hypoxia factors. So that's how the direction of growth or development of new blood vessel also will be affected towards the place that actually those cells that induces the first primary hypoxia factor. This is good for research study actually, as a nice topic. Okay. I want to inform you that about hypoxia, which is a kind of primary stage of informing and in a primary stage of angiogenic formation, especially in tumor development. At the kind condition that the oxygen is normal around the cancer cells, the blood vessels doesn't produce any HIF factors, which is hypoxia-induced factors. They won't be that much over there, and then it's okay for chemotherapy, susceptible to chemotherapy and radiation therapy. But when hypoxic cancer cells happen, the area around cancer cells are low in oxygen. In that case, the genetic instability will be increased. So the gene will be not that stable, and immune system will reduce. In that case, you will experience a high expression of hypoxia-induced factors or HIF factors, and then angiogenesis will modify. That's a reason that it means that it will stimulate or it will change the direction of angiogenesis toward angiogenesis, not anti-angiogenesis. So it's a kind of guidance to change the system to increase the number of blood vessels at the direction that hypoxia is happening. Okay, I talked too much about angiogenesis. Now I want to introduce you some other phenomena, inflammation that happens. Almost 20% of cancers will be linked actually will be started from inflammation. And there is a correlation between inflammation and angiogenesis in cancer progress. So interesting part is the markers which are which are contributing in the phenomena of angiogenesis and inflammation, they're almost the same, you know. That's why it will affect each other, and then almost they are similar in the pathway. So you can see here what will happen. As you can see here, the tumor cancers, the cancer cells, actually the cancer cells will the cancer cells will produce some growth factors like VEGF, which will take out, infiltrate actually monocyte or macrophages to tumor area, will bring it towards itself, and then will educate it, you know. It will educate it. It means that it will make some changes on it. It will induce some modification on it and convert it to another molecule, like another type of macrophage, which is called a tumor supportive or tumor associated macrophages, or TAM, you know, TAM. So this factor is actually the factor that can induce angiogenic factors. That's how that induce protease like metal protease to go and then degrade the membrane, the basement membrane, and start invasion. That's how the start of angiogenesis will happen. So actually, the series of events in order to start new blood vessels to get more nutriented oxygen is starting from these tumor cells that by secreting growth factors can modify monocyte or macrophages to infiltrate the tumor area, educate it, modify to convert to TAM molecules or TAM macrophages. And this TAM, which is educated by tumor cell, will induce factors that can cause angiogenesis to happen. So some cellular pathway will happen also to induce inflammation, and that's why most of when you design some compounds to have anti-cancer effect, if it has anti-angiogenic effect, mostly it has anti-inflammation effect as well. Okay. As I said here, if you look at here carefully, it was TAM macrophage that induce angiogenesis, right?
Muhammad Aidil Syai.
I would really thank. Okay. So here, using this phenomena affecting this TAM, I mean, using based on these TAM macrophages, some drugs has been designed like clotronate that was encapsulated in liposomal form of molecule, and as you can see here, it was injected, and then the phenomena of administration of clotronate was just inhibiting these thalamic macrophages, so it could not go further, and then could not increase the growth factors to cause angiogenic process. So the ultimate reason was stopping angiogenesis, so making the cancer cells to go toward being toward a starving condition, and ultimately cause death in cancer cells. That's how, you know, such mechanism of treatment happened based on this inhibiting this TAM. It was just one of the types of the treatment. Okay, I told some briefly about cancer events, and then some mechanisms that can make it to happen or progress and develop. Now I want to mention some in vitro analysis in cancer research that it will be quite useful for students under further studies. It's good, you know, for you to get some idea about how to test, how to start actually in this field. So I thought maybe it's better than other details. For example, if I mention here. So first of all, when you have a molecule or agent synthesized or extracted or whatever, and you think that it has anti-cancer effect, first of all, you need to check the cytotoxicity potential of that agent or molecule, right? So MTT assay is a kind of colorimetric test that based on the agent called MTT, it's a kind of colorimetric, recognition. You can identify the percentage of the cells that has been killed after being exposed to a specific concentration of your agent. So how is it? It's actually, you can just see it in 96 cell players. You can see here, you can see your own specific cancer cell line here, and then in a specific, right, actually media. After that, you will treat it with your sample, and then after that, you will expose it to MTT, which is yellow in color. And then after 3.5 hours in incubation, you will take out, aspirate all the media, and then we'll add some DMSO. Something will happen. DMSO will convert yellow color MTT to purple formazan, and then based on this colorimetry, you will see that using a spectroscopy, you can find out how much the percentage of the cells has been killed after exposure to your molecule. But the mechanism of MTT is like this: that MTT is actually kind of soluble molecule that, after being exposed to mitochondria of live cells, will be reduced to purple formazan. Formazan is a kind of insoluble salt. This insoluble molecule then will be solubilized using DMSO and will convert from yellow color to purple color. That's how you can see. So the most bright veil that the cells has been killed the most here. For those, for example, negative control, which hasn't been exposed to any treatment, all the cells are alive. It means that it will be like quite purple color. Okay. Another test is colony formation assay. If you want to know, for example, as you can see, I'm step by step explaining different stages of angiogenesis that I explained, which is needed for new blood vessel formation. So in colony formation, you will see it in petri dish, sterile petri dishes like this, a specific number, very low amount of the cell line, a specific cell line that you are going to do some research on it. And then after that, you will expose it to your agent or drug that you want to study. And you can see, for example, if it is really effective agent, then the number of colony will decrease. As you can see in A and B, you see that the number of colony is very less compared to negative control. They can see here, you can see plenty cells gathered together, divided here, and then as a big colony. This can be considered as negative control without any treatment. But as you can see here, the colony has been reduced in number, so that's how you can estimate the colony formation inhibition potential of your drug. Okay, another stage, as I said, is migration assay, right? So in migration assay, also it's very easy to do. You will see your plates, with plenty of the cells. They will fill the whole bell, and then after that you cut or make some injuries, as you can see here, and then after that you treat the valves, and after individual time, a specific time, actually you can see that, for example, the negative control will start to close because the cell has a potential to migrate fast and close the injury. But actually, when you treat it with your drug, you see the speed of closure will be lower compared to negative control. It means that, for example, this drug has this much percentage potential to inhibit the cells from migrating. It cannot be migrated if you induce this drug. So, okay. Tube formation. Tube formation is the methodology studies the differentiation step of angiogenesis. As you can see here, when cells, start to migrate, and then after that, meanwhile proliferated, invade, and now they start to differentiate in morphology. How it will do is actually using some growth factors. It will start to make some tube-like structure. So if no drug happen here in normal condition, yeah, here you can see the tube-like structure will be normally prepared very nicely and then without any breakage, you know, and then the area inside them also will be counted using some software like ImageJ. But if you have exposed it to your drug, which is quite effective, or maybe high dose you have used, then no tube-like structure will happen here. You see? So this is kind of we consider that compound or drug or agent as a compound that has inhibitory effect in differentiation stage. I mean tube-like structure or tube formation stage. So if you want to compare this slide with this one, you can say, for example, it can be the same drug, but with higher dose here in part C, and then but lower dose in D. So if you increase the dosage, it will ultimately reach to something like this. So this is something, you know, you can estimate the percentage of potential actually of the agent, how much it can inhibit the formation, tube formation, how much it has the possibility to inhibit going towards a differentiation stage. Hang drop. This is another study that I'm sure that you will enjoy if you do it. But it is a kind of new method. You know, it is very costly to go through tumor model studies if you want to see whether your sample, your extract, or whether your compound has the potential really to work on tissue or only is effective in cells, you know, because tissue is a colony, gathered colony of the cells in three-dimensional condition, right? So hanging drop is an easy assay that will give you chance to try your sample in three-dimensional condition. As you can see, you will induce in a viscous media, you will induce your cells in viscous media and let it to grow in three-dimensional condition. After that, you will seed them and you will treat it with your sample and compare with negative control. So after 6 days of treatment, you will see that, for example, your negative control will grow very fast. You know, you see it's grow very big because nothing has inhibited from growth. But meanwhile, the sample that you treated, you see the size of the drop, which will be considered as a tissue, doesn't grow too much. So it means that this sample has the potential to inhibit this kind of cell line in a tissue form. So hanging drop assay actually, due to this reason, can be considered as in vitro tumor model. Okay, this is very economic test actually. CAM assay is very economic test because you just need, you know, some eggs and you can do it, but in a sterile condition. In this assay, as you can see, you need to just open a part of windows on the egg and then expose your medicine, I mean your drug or compound, whatever, and then put it back again and wrap it properly and put it in the proper condition incubator. And after a few days, like not few days, after 2 weeks, you will take it open and then we'll monitor it under microscope. And you will see that, for example, for negative control, you see plenty of blood vessels happen here and move forward. But compared with the sample that you did the treatment, you won't see that much new blood vessel form. So it means that your sample has the potential actually to inhibit the formation of new blood vessels. And it's another test that you can estimate how much your sample is effective in inhibition of tumor in angiogenesis step. Okay, well done. Until here, you could do numerous in vitro tests. And then here you will get some idea about, you need to get some idea about in vivo. You want to get more idea. You want to see in reality what will happen in the body. Okay, in this case, you need to go forward using some animals, whether nude mice. Nude mice is, very popular tumor models in cancer research studies because they are modified, genetic modified, and then they don't have immune system, so easily can absorb, cancer cells, and it can be, we can induce tumors easily in those kind of mice. But after this, some melanomas, some tumors like melanoma can be induced even in albino mice, even though they have immune system, So it can be happened. We can develop some tumors in other animals like guinea pigs, even dogs, monkeys, rabbits, even, But don't forget to get ethics for them, and behave humanly with the animals. Don't let them, go through pain, and love them. Animals are more important than human models, even though. So after doing such tumor model studies, mostly the tissue will be taken out and sent for histopathology studies to see what's happened exactly after your treatment in the histology of the tissues, or what happened to biochemistry results of the blood, animal blood, and you can get so many data about your drug. I just want to tell you briefly, very small thing here is about how to induce the tumor. Some researchers that believe that easily they just inject the tumor, to the animal. I mean, inject the cell lines to the animal and create tumors. But when I was at USM, I faced some supervisors and then actually some supervisor and researchers that they don't believe in inducing the tumor by injecting the cell. They said that, for example, when you induce the cell, it doesn't mean that it happens the same in human body. So what they do, they use some reagents and some carcinogenic agent, and then they let the animal to go through the process of tumor formation in the body quite naturally under exposure of such agent. They said that inducing the cells to the animal and making a straightforward the tumor is not that much beneficial. That exposure of the animals, to those excipients, or agent can cause tumors. So they said that it is more accurate. This time is more accurate than straightforward inducing the cancer cells. So it is something that can be considered. Sometimes you need to run the study and you don't have time for that because so many other parameters will be out of your control. So it has its own pros and cons. So you can go ahead. Depends on your time, depends on your criteria, and so on. Okay. I want to use the time here now and talk about some cancer therapeutic strategies. Until now, which kind of therapeutic strategies have been employed? First surgery, some tumors, they are risky to continue going towards chemotherapy. So it's better to do surgery and remove it all at once. So in that case, then going through surgery is better option. Something like melanoma, which is quite aggressive type of tumor. If you go through chemotherapy, it will definitely metastasize to other tissues, and it will be difficult, to survive. But in that condition, immediately they do surgery, remove the tumor because it's just on the skin, or maybe deeper layers of the skin, and then remove that area. After that, probably depends on the physician. They will go through some chemotherapy or probably radiation therapy. sorry. Sometimes the phenomena is like this, that therapeutic strategy will be combined with each other. For example, sometimes, for example, the patient is having too much pain and can't control the pain. In that case, we'll go through, the doctor will advise him to go through radiation therapy. Radiation therapy is a method actually in order to shrink the tumor size fast, actually within, like 2 weeks or 1 month, very fast. So the pain will relieve the pain. So sometimes when the doctor wants to advise to do chemotherapy, first we'll ask, okay, go and do some radiation therapy, some sessions. After that, we'll go chemotherapy. Or sometimes after surgery, they will say, okay, go to chemotherapy. And meanwhile, you can, after finishing chemotherapy, few sessions of radiotherapy also will be advised. So depends on the condition of the patient, it depends. Sometimes combination of all the treatment will be needed for some patients, but sometimes just one or two methods will be okay and the patient will survive. And another method that I will describe more here is about immunotherapy. In further slides, I will explain it. Okay, chemotherapy drugs are actually synthetic drugs that they will affect different phases of cell cycle. So mostly synthesis phase and mitosis cells, mitosis phase. So these chemotherapy drugs, they are not targeted for a specific target, they are not smart molecules. That's why mostly they have side effects in different person. Actually, it is quite different. Phenomena will happen after being exposed to chemotherapy. And sometimes, for example, when the immune system will decrease, the tolerance of the patient also will decrease going through chemotherapy. I think it is quite painful. Okay, radiotherapy is exposing the patient, the tumor of the patient, actually, towards to X-ray, gamma ray, or proton beams, to shrink the size of the tumor. Sometimes it's quite urgent, Sometimes, for example, some tumors will happen to something like sacrum bone, which will make extra pressure on spinal cord. So the physician doesn't have any other choice. Just he goes ahead, using radiotherapy to shrink out the size of tumor and take the pressure out from spinal cord. That's the only choice they can do in a short while. So it's a kind of asset. This method is a kind of asset for combining with other methods to make the patient more relaxed during the treatment. Okay, targeted therapy is a new kind of system of drugs for cancer therapy. How they do is actually like this. They take out, they estimate or identify a specific targets that can cause cancer, in different types of cancer, and then they design a kind of drug that can target specifically those targets. So that's how it will inhibit the cancer cells by targeting only one specific molecule. That's how we call it targeted therapy. It's better, than compare with chemotherapy, because it targets only one molecule, specific molecule, so it doesn't affect other normal cells. So it won't be as painful, or it won't have such side effect that chemotherapy has. And that's because of being quite straightforward, targeted towards the specific molecule. It can be produced as a chimera. It can be, can be produced in human or other animals. And one example is, targeting VEGF, which is a vaccine. This technique actually is much advanced than conventional chemotherapy in cancer treatment. So, okay. Anti-cancer phytoconstituent. I want to explain more in this slide, because actually during my master and PhD, I worked on some phytoconstituents, and then their effect in cancer treatment. You know that most of the plants, especially the plants in Southeast Asia, those plants have are high in flavonoids, and then that's why they have anti-cancer effect and antioxidant effect. So, but as you know, those flavonoids which can have potential to inhibit cancer from progressing, but it won't have that much effect in vivo, because they have a very big ring, cyclic ring, that they cannot pass through cell membrane and reach to the cells. That's why it won't have that effect in vivo. So what the drug delivery had them in this field was encapsulating those phytoconstituents, or a proper standard extract of those herbal extracts, in the form of lipidic form of vesicles, which has different conformation, like liposomes, like emulsions, like phytosome, or even etosome. Each one has their own stories. I don't think I will get enough time to tell all of them about them, but it's quite interesting, because liposome was a kind of primary form of vesicles that would help the phytoconstituent with very high potential for anti-cancer, as an anti-cancer agent, to move towards the human body, especially, or animal body, and could have proper, even comparable with a standard drug in vivo. So another type, for example, etosome, that I worked this type, during my PhD, which is quite high in ethanol content. This kind of vesicle is very interesting, because it can shrink itself. It can shrink the vesicle and then pass through different layers of the skin. It's a specific vesicle for topical application of phytoconstituent. It's good, to read more about this kind of vesicle and the applications of such phytoconstituent, that who knows, maybe they can have better cytotoxic, or not cytotoxic, better anti-cancer effect, in vivo, compared with a standard approved drug. Okay, one of the companies that I was actually a scholarship attached in my PhD to, this company was Nutraceuticals. So in Nutraceuticals, what they do, they did very interesting part of the job. They did, they planted the orthosiphan estaminus. You see, I'm sure that you know about it, because it's quite famous over there, even in Indonesia. They use orthosiphan estaminus as a base plant, and then they planted, in a vertical plantation, and standardized the extract, and use this extract, which is quite a strong and anti-angiogenic effect. But the issue is they developed a specific formulation in order to enhance the delivery of phytoconstituents, which is effective in anti-cancer and then anti-angiogenic effect. So they developed a novostatic sample as a sachet, and then the factory is still in Malaysia. And they're selling the product, and it is as a food supplement for time being, but it is in phase 3 of clinical trial for breast cancer and colon cancer. And I heard that they are in phase 2 of clinical trial against COVID-19 too, because it's quite high in antioxidant efficacy. So what I did during my master, I developed this formulation, Cancer 5 Nano, which is commercialized now under Nutraceuticals, and is under sale in Australia and Malaysia. Cancer 5 Nano is actually a specific modified type of liposomal formulation using orthosiphan estaminus extract. This is using quite a standard extract, and then it enhances the efficacy of the primary extract to 3 or 4 times more stronger. So I'm sure that outside over there are plenty of researchers working on other herbal extract that wish them work harder and then get better results. Okay, microarray. Now I'm moving forward to immunotherapy, another therapeutic strategies in cancer treatment. But first of all, I want to share some brief description about microarray. Okay, what is microarray? Microarray is actually like this. It started like this. The best way for a physician to start taking a therapeutic strategy regarding to patients is to get more information about what is the exact type of cancer this patient is tolerating. So even though the standard method until now is going to biopsy and then finding out the type of cancer, but that kind of studies doesn't give the physician exact health information about, for example, how much is the rate of proliferation, or for example, how much this patient, this kind of tumor has the potential to be resistant to a specific treatment. You know, this is something that you won't get any information using biopsy. Or for example, what is the possibility of this tumor in this patient to not to answer to specific treatment, or how much is the rate of progress. You know, these are detailed things that may help physicians to employ proper, actually, and appropriate therapeutic strategies. So in order to get such information, microarray will help. How it can, it's a kind of chip, you know, which is specific for proteins, DNA, and RNA. Actually, what will happen, for example, if we have abnormal protein in a patient, we will have a kind of probe in our chip, microarray chip, and which is tagged with fluorescent dye at a specific parts of the microarray chip. As you can see here, for example, like DNA microarray, you can see those areas, those dots that are not, those dots that are not present in the cells will be shown as a gray color, and then those in pathological cells only which is available will be which is considered as mutated kind of genes will be shown with red color. Only is available will be shown as green color. So using such template, it means that comparatively with a normal condition, you can identify the abnormalities in whether gene, DNA, or protein. If you want to study the protein, you know, sequences or any abnormality in proteins, then you need to have a protein microarray, which is composed of, you know, some antibodies, probes that is connected to that is tagged actually with fluorescent dye in a specific part. So based on such methods, you can identify the abnormalities in protein expression of your patient compared with normal healthy condition. Okay, here I can see that, for example, this much data in just less than few hours you can get for genome detecting, so you can find out any abnormalities, you know, in DNA, or whether here you can see for variance actually in protein expression in your patient. So that's the new tactic actually that will help more during immunotherapy. I will explain more in the next slides. Okay. One of the methods in immunotherapy is using CAR-T. CAR-T is actually a chimeric antigen receptor cell therapy. It's like this, briefly, if I want to share it with you. Actually, they will take out, they collect blood from the patient, and then will isolate T cells and reprogram it to be more effective. You know, will boost the efficacy of those T cells, and then we'll target, you know, some we'll use some targets like HLA, human leukocyte, antigen, and then design a specific receptor and then induce it to the patient again. When they induce it to the patient, those targets, those receptors, we call it as CAR-T, will connect to the antigen on the surface of cancer cells, and then inducing such modified T cell will go ahead and then attack it quite fast and then efficiently, and will kill the cancer cell. This is just a simple vision of CAR-T, even though, you know, it was very successful, very interesting in immunotherapy at first, but since the HLA is not the same in all human beings, if in each individual is different, that's why in some patients it doesn't give good response. And also it's not effective in solid tumors too. So another technique which is called as precision medicine, which just started after 2010. What they do is like this, you know, it's using some omics system. What does it mean? As you can see here, you see previously traditional medicine was like, for example, they designed a vaccine. This patient, all of them should use a vaccine without considering, okay, this individual genetically is quite different with this one and this one, but all of them is using the same drug. But in personalized in personalized medicine, based on each person, their proteins, their genome, and then their exploration of the genome, the designation of the drug will be quite different. That's called the kind of personalized medicine. Okay, what they do exactly, this is like a this technique is a kind of ocean, you know, because it's a kind of collaborating between metabolome, transcriptome, epigenome, microbiome, proteome, genome, you know, all these fields will be considered all together, and they collect as much data as possible. They collect, for example, urine, they collect blood, and then from the patient, and they analyze a protein pattern, and also gene expressions, and then a modification of the patient all together, and find out what is the abnormal part. So when they find out the abnormality in those patients, they will do something. They will synthesize the right protein, the correct protein in the lab, and then at a specific time intervals, they will inject those synthetic proteins to the patient, even though, you know, before that the patient should go through some chemotherapy to reduce the immune system in order to get it done, you know, to get it to accept those synthetic proteins to work in their body. So it is something like this. So after, for example, a gradual injection of those synthetic proteins which was lacked in the patient, it is like a hit that the genome of the patient will start to learn, you know, okay, if this is the right protein, it's here available. So the correction will happen gradually in the body. Even though sometimes it works within like 17 months injection, you know, in some patients, but sometimes it doesn't work properly. So probably it needs to be repeated for other sessions, other duration of time. But it's quite new area of the cancer treatment that some research groups in US and recently in Australia, they are going ahead through them. And I'm one of the scientific advisor states in Australia that is working on clinical trial of this kind of studies. Okay, but I told you about in this slide about CAR-T, right? Just remember this. And now I told you here that the target that they selected in this method was HLA, okay? newly discovered T cell. This is, the recent news that happened in as advancement in immunotherapy just recently in 2020. I think in January they published the news that they published the article that they found out another target, which is MR1. They found MR1 is a better target rather than SLA because it is, it is something that is the same in cancer cells in all individuals, not only a specific number of the human being. So it's easier to target it, and then they synthesize the receptor for this MR1, which is called TCR. So they take out the T cell. They isolate T cells, they modify, boosted the function, and then by injecting the modified T cell along with TCR, they could this TCR could tag with, could bind with MR1 on the surface of cancer cells, as you can see here. Okay, so this binding can be a kind of alarm for T cell. Okay, go and kill the cell and will attack the cancer cells efficiently and finally kill the cancer. So this is actually the latest advancement in immunotherapy, the field of cancer therapy. So I hope that you enjoy it and one day you find the next step. Who knows, maybe you complete this day. Thanks a lot for your attention. Okay, if you have any question, you're most welcome. Hello.
Hello.
Yeah. If the audiences have any question, you're most welcome.
Okay. Thank you to Dr. Mansoureh for giving us knowledge and information about this topic, cancer. We are continuing our event to the next question and answer. If you have questions related to this topic about cancer, feel free to ask us on the chat section or raise your hands. Our team will help you. To all the participants, if you have questions, please feel free to ask on the chat section.
Yeah.
Okay, we have one question on the chat section from Ranti Yulia Marnis from Sumatra Institute Technology. She asked, in cancer patients who are undergoing chemotherapy, good and bad cells will die. Then will nerve cells die too? While nerves cannot regenerate, is it dangerous if the nerve cells also die? And for end-stage cancer patients who are undergoing chemotherapy and the effects of nausea.
Sorry, the voice is disconnecting. Can I see also? Is it like text?
Yeah, it's on the chat section.
Okay, wait. I should find. Here.
Yeah.
Okay, you can read again, please. Yeah.
Okay, first question from Ranti Yulia Marnis from Sumatra Institute of Technology.
In cancer patients who are undergoing chemotherapy, good and bad cells will die. Yeah, of course. Then will cells die too, while nerve cannot regenerate? Is it dangerous if the nerve cells die too? And for end-stage cancer patients who are undergoing chemotherapy and effects of nausea and vomiting or extraordinary. What is the good handling? Okay, actually during chemotherapy, yeah, since we don't have individual targeted therapy, it's a kind of synthetic drug that affects, all each and every cell. So anything can happen. Sometimes some cells will die, normal cells. That's how that, for example, some patients they lose, they cannot control their urine, or sometimes they have very difficulties in other normal works. It is not like this that definitely will lose all, because some of them can come back, some of those abilities can come back, like hair loss, but some of the effects, some of the side effects are not returning back. Sometimes, for example, if any nerve be affected, definitely it won't be regenerated again. So, but what you can do, this is something that cannot be avoided, in a chemotherapy. So for fatigue, yeah, something like nausea, vomiting. Actually, I suggest actually using, herbal products that are having less side effect. Even though I cannot advise, I'm not physician doctor, I cannot advise clinically for cancer patients. But based on my knowledge, whatever I experienced in other cancer patients, it was easier for the patient to tolerate, these side effects like fatigue, while using herbal standard products, not whatever they find, for example, like herbs, go and eat. Standard products means proper formulation of products which are based from herbs, and then they don't have as worse side effects as synthetic drugs has. Even though it needs to be checked with the physician, because sometimes it can damage or eliminate actually the effect of those synthetic drugs, which is called as chemotherapy. So it's better to check with physician and don't use by their own, anything. I read one Google that chemotherapy is usually given in a count of cycles, which consists of period of rest. For example, chemotherapy for 1 week, then followed by a rest period of 3 weeks. My question is it possible for this chemotherapy cycle to change depending on how worse the cancer is? Well, no, actually, how the physician decided, it's actually dependent on the ability, the tolerance of the patient, depends on how is the physiologically condition of the patient. Some patients can tolerate, continuous cycles and shorter cycles to rebuild the cells, actually, because it meanwhile, even though it's attacking the cancer cells, meanwhile is attacking, killing the normal cells too. That's why it will be sometimes out of control for the patients. So it is not like this that if it is worse, so continuously make the cycle longer. It depends on the condition, physiological condition of the patient, whether it can be tolerated continuously chemotherapy or no. Otherwise, we don't want to keep the patient. We are trying to make them survive.
Okay.
So yeah, I don't know. Did I answer their question? I think something is written here. GK, you saw. Was it question also or no? I have some question. Cannot talk. Okay. Yeah, sure. I want to write here actually my email address. You can forward me if you have any question further up, or if you need any guide, any consultation for running running your studies, to design actually your cancer research, or if you want to continue your studies in this field, feel free to contact me. I wrote my email address here.
Okay, thank you so much, Dr. Mansoureh. Next question will be
Yeah, it's
from
fine.
Dimas Pangestu. Please to Dimas Pangestu.
Good afternoon, Dr. Nazari. My name is
Hello.
Dimas
Hello.
Pangestu.
Okay. Nice to meet you.
Yeah, nice to know you. The question
Oh, thank
is,
you.
in thinking about in thinking about stem cell interaction with the tissues as a possible cause of cancer, the basic idea of DNA dynamic is relevant on the opposite side on the metabolism. DNA is involved in producing the molecules that interact in the tissues system and regulating the cells in the tissues. But it is interesting to wonder which side of the metabolism causes cancer. It is the DNA or the cell interaction.
Got it. You cannot identify, you cannot differentiate it. Cancer is not, it's not one mechanism called, okay, that's cancer. It's ultimate, actually, it is a kind of collaboration between the whole system. I think you are talking about ADME, right? Different stage of mechanism and which stage you should study in order to find out. Okay, we studied, for example, cancer, whether it is in cell signaling or genome. Am I right? Did I understand properly? Did you mean this? Yeah. It is not like that. You need to actually study the whole phenomena. But since we are not able to do that, it is something that you can do it in a team. For example, one part of the team will work specifically on cell signaling. The other part will work specifically on the other phenomena. But all together needs to be done properly, and then that's how the multidisciplinary has been invented since a few years back. They are studying, in a teamwork, combining all the information from genome, cell signaling, bioinformatics, and then so many like proteomics. That's how they started now working since 2010. They're working like this on the study. But if you are talking about how to do your study during your master PhD, I one part and then go ahead towards that field. But you cannot claim that you for example entered cell signaling, so you know everything about that type of events. It's not like that. I don't know, did I reply you properly? Or if you have any further question, you can ask.
Oh, okay, thank you, Dr. Nazari. I'm interested in how stem cell interact with tissues and how those tissues normally metabolism.
Oh, the stem cells. The stem cells actually, I did work on a stem cell. That's why, because I worked more in herbal products and then working on molecular signaling. I didn't go through a stem cell, but as I know, stem cells can work in a specific tumors, like if I'm not mistaken, in myeloma, multiple myeloma, and then leukemia. But it's not working for all types of cancer tumors. Since, for example, for solid tumors, you cannot use this system. It will happen like this. In cancer, in leukemia, for example, your blood cells will be quite modified. It will be abnormal. In that case, what will happen? You use another healthy the stem cells and will induce it to the patient. It will stop production of new blood vessel, even though it cannot be acceptable for each and everyone. It has quite a costly process, and it's not possible for each and everyone, and it's not 100% successful. It is limited, and it is limited for only leukemia and multiple myeloma. If you want more details about it, actually I didn't work on it, but if you want, I can join you to a group later on that may help you in this. I put my email here. I can do this for you. Probably you can get better consultation with that group. Is it fine?
Thank you,
Hello?
doctor. Hello.
You're welcome. Yeah,
Thank you,
sure.
thank you for the answer. I hope you have a great day. Stay healthy. Thank you.
Thank you, me too. I guess Tina would like to ask how much influence does stress have on cancer? Okay, yeah, good question. How much stress can have influence on cancer growth? Actually, stress can decrease the immune system. It means that when your immune system is down, so the T cells, which is as I showed you in immunotherapy, your T cell number will decrease, so they won't be efficient enough to attack cancer cells and then kill them. So stress can directly decrease your immune system against cancer growth. So growth will be faster, and then it won't respond properly to the treatment, and mostly cancer will die.
Okay.
Yep.
To the next question from Suri Isnaini on the chat section. Hello, Dr. Mansoureh. How are you doing? I hope you're doing great. I would like to ask a question. Could you tell me what is the important things we as a pharmacist should tell to the cancer patients during a counseling on their first chemotherapy?
Yeah, okay. Good question. Actually, first of all, you know that in cancer patients, once they understand about their disease, they feel quite disappointed. They just imagine their own death, and they don't think about being survived at all. So first of all, think about holding their hands and emotionally, understand them, and then make them have hope in their own survival, because the feeling that they have is actually the first step to decrease the anxiety, to decrease the stress, and think about the improvement in their condition. So after that, I believe that first of all, the actually emotional feeling, the internal feeling of the patient is the most important help that the pharmacist can do. I mean, a physician especially can do facing a patient. And then collecting proper information. Proper information means proper data. Don't justify immediately for the therapeutic strategy. It can solve actually with a specialist. What happened? Hello, what happened? Yeah.
The next question. Hello.
Rizky Hidayah. Yep. Yeah.
Okay, to the next question. For the participants who want to ask, please feel free to ask us on the chat section or raise your hand.
Yeah, if not, all is okay. I wrote here my email address. You can just drop an email even further. If you want to design your study in this field, you know, it will, it will make me happy to give any consultation in designing research.
Okay.
Excuse me, can I ask?
Okay. Please to ada sebagian Maulana.
Yeah.
Yeah.
I have.
Okay. Thank you, Gena. Hello, Dr. Mansoureh. Yeah. I have read.
Yeah, fine. Thank you. How are you?
I have read an article that the CRT cell have some adverse effect, and one of the adverse effect is viral infection disorder. And my question is any possibility people with cancer who has therapy with the immunotherapy with CRT cell should take an antivirus for prophylaxis?
Sorry, I didn't understand because it was in the middle of your sentence was disconnected. Can you? Chemotherapy with what? Chemotherapy with what?
Hello. Maybe you can type on the chat section to help Dr. Mansoureh.
Okay.
Yeah. Yeah. How effective as stem cell for cancer treatment? Stem cell can be beneficial in cancer treatment, but not for get some advantages in treatment. For a specific type of cancer, not solid tumors, for myeloma and then myeloma and leukemia. So sometimes we need, you know, combination therapeutic strategies, not only one. Yeah.
Okay. Okay, actually, Dr. Mansoureh, I have a question while we're waiting Ade Septian. So I had a relative that had a breast cancer, and doctor told their daughters to do some tests. So is cancer hereditary or not?
Okay. You know, there is a still discussion on it. I said that it's a kind of genetic modification actually that can be influenced by environment. It means that, for example, a patient the current patient may have some modification in their genome, so it can be inherited from the last generation, but it could be not happen, you know, due to environment too. If some condition like exposure to some chemicals, you know, or some sadness feeling or anxiety or stress, continuously stressing life, this can affect environment that can affect the expression of those proteins that can cause the series of cascades and ultimately reach today cancer. So we cannot say straightforward it is inherited. There is possibility of inheritance, but it's not everything. You know, the style of life it matters really, but inheritance also matters. But it can be controlled. That's how, for example, nowadays they do, you know, some genome analysis in patients, individual patients, to find out, for example, what is the percentage of each individual to get some cancer. So, but they don't say 100% this person can get cancer because her mother got cancer. But if the lifestyle promoted, yeah, definitely it will happen because it's more towards that area.
Oh, okay. Thank you for your
Yeah.
answer. Okay, last question.
Yeah,
We
you're
return
welcome.
to other
You know what? Sorry,
chat section.
I want to complete my sentence with another issue.
Okay.
When I was in Malaysia, in the company that I was joined as a scholarship student. They were advising the family members of the patients to take some anti-cancer drinks, on daily basis to prevent actually the occurrence of cancer in the future. So some antioxidant drinks will help will help the people from experiencing cancer in future if they use it. Actually, they take it on daily basis, but not every sample. I'm talking about a specific type of research and studied formulations like the samples that we prepared, we studied on it, so many articles and patents has been published about such food supplements can help, as a daily based multivitamins that you're taking. You can take, some antioxidant samples as a food supplement drink, like whatever you are drinking daily. It doesn't have side effects, but at least you are doing your best not to experience bad about cancer. This is something that we are also as a science doing.
Okay.
Doctor, maybe I have one
Yeah.
question again. Maybe this is
Yeah.
not have significant relationship with the cancer, but I have question. During COVID-19 infection,
I
antibodies
hope to be able to
are
answer. Yeah, okay.
okay. During the COVID-19 infection, antibodies are developed against virus. Is these antibodies are lifelong for the strain competing against infection? What do you think?
A strain what competing against infection? What?
Sorry?
Can you repeat again? Can you repeat your question again, please?
Oh, okay.
Oh
Okay.
my God, the voice is horrible.
Yeah. During COVID-19 infection, antibodies are developed against virus. These antibodies are lifelong for the strain competing against infection. What do you think?
Of course, no. Of course, no. You're talking about COVID-19. The genome is modifying every day. Within just 8 months, more than 200 mutations just happened in the genome of this virus. It is not something like that. That's why it's not easy, to talk about it or decide about it or think about it. But I don't think so. It can happen like this. There won't be like a specific antibody that will be effective at long life, because it's modifying. It's not stable virus. It's a kind of educated virus actually. New seven logics. Actually, it's good, to share this with you. Logics is a company in US. They're working on precise medicine. What they're doing is taking out the antibodies from each individual, because the genome modified in each individual is quite different. So based on that knowledge, they are trying to even work on medicine for against COVID-19. Doing in this research in US currently, but they cannot design, same drug for all individuals. They are doing it only. It can be gained only for each individual one by one. For example, the medicine, the antibody that they are designing for me, it will be quite different with what they are designing, for example, for other patients. You know, that's how the precision medicine is called. So what I guess is something like this. Even I'm not sure about the vaccines too. Even though they inform that the vaccine is in clinical trial level, some of them has been rejected. But whatever vaccine you design, if the genome is changing, it means that the vaccine is quite the virus is quite small. It will find a way to go out of the step and then find another way, so the previous vaccine won't be effective on the new generation of those viruses. So that's why I think the best way is increasing the immunity system of the human being. It's better to work on immunotherapy against such type of virus. That's how they are doing like plasma therapy nowadays, you know, in